Rebecca Skloot, born on September 19, 1972 in Springfield, Illinois, is a science writer who has published over 200 featured stories and essays and wrote The Immortal Life of Henrietta Lacks, a #1 New York Times bestseller book and a critically acclaimed novel. Henrietta Lacks, born on August 1, 1920 in Roanoke, Virginia, was an African-American woman who was unknowingly the donor of cancerous cells from the tumor in her cervix. These cells taken from her became the first immortal cell line known as the HeLa cells and were used for research throughout the 50s up until now. After finding a lump on her cervix through a self-examination, Henrietta decided to go to Johns Hopkins Hospital, the only hospital that would provide care to African-American patients in the 50s. Her doctor, Howard Jones, took a biopsy of the mass and sent it to the lab to be examined. After learning that the biopsy showed stage 1 cancer of the cervix, Howard Jones's boss, Richard Tellind, decided to send the results to George Gay, the head of tissue research at Hopkins. Gay was looking for a way to grow the first live human cells outside the body. Up until Lax's cells were cultured in the lab, all of the previous cells had died. However, 2 days after Gates' assistant, Mary Kubasek, put Lax's cells in the incubator, they began to grow uncontrollably, 20 times faster than normal cells, becoming the first live human cells to be immortal. After growing the first immortal human cells, Gay told a couple of his closest colleagues about this phenomena and was asked by several of them to share the cells for further research. Gay said yes and sent the cells around the world, making HeLa cells the first living cells to be shipped in the mail. Henrietta Lacks died on October 4, 1951 at Johns Hopkins Hospital at age 31 from an aggressive strain of cervical cancer. An autopsy showed that the cancer had metastasized throughout her body. She was later buried in an unmarked grave in her family's cemetery. In the 1950s, Jonas Salk used HeLa cells to create the first polio vaccination due to the large public health problem that had been occurring. He needed a large bulk of cells to study and test his vaccination on, so a factory opened in 1953 to mass-produce HeLa cells. This factory was started at Tuskegee University in Tuskegee, Alabama, and provided scientists with HeLa cells they could do research on. At first, the lab operated as a non-profit organization, but as demand grew higher for HeLa cells, they began to make a profit off of them. In 1955, Philip Marcus and Theodore Puck cloned HeLa cells at the University of Denver and became the first scientists to successfully clone human cells. The cloning of HeLa cells allowed for advances in science fields such as human genetics and in vitro fertilization. In order to guide reporters away from finding out Henrietta's identity and who the source of the HeLa cells belonged, George Gay came up with the pseudonym Helen Lane. Gay did not want to release her name because of doctor-patient confidentiality, but in the end, everyone found out. Chester Southam, a virologist working in the field of cancer research, wanted to see if HeLa cells could cause cancer if injected in other patients. He began his study without their consent and was later found guilty of unprofessional conduct by the Board of Regents at the University of the State of New York. Stanley Gertler, a cell and molecular biologist and human geneticist, proposed that HeLa cells had contaminated numerous cell lines. Through his research in examining isoenzymes in 18 independent cell lines, Gertler discovered that they were all identical, which suggests they branched from the same line. Following the death of George Gay, Gay's associates wrote a tribute about him which included the famous HeLa cells. After going through the research, they discovered that the donor of the cells was Henrietta Lacks. In the tribute for Gay, they included Lax's real name, and for the first time, it was written correctly in print. Researchers were so intrigued by Henrietta's cells that they wanted to do more research. Without informed consent, they took cells from Lax's children and studied them. This became one of the biggest ethical issues surrounding Lax's case, as neither her cells nor her children's cells were taken with consent. Informed consent is permission given by a patient to a doctor for treatment with full knowledge of possible risks and benefits. This law was implemented in 1974 and was required to be acknowledged for all human test subjects after it was not done so for Henrietta. A Rolling Stone article was published about Henrietta and her family without their knowledge. In the article, it states that HeLa cells were being sold for a profit, and when Lax's family found out about the article, there was nothing they could say to stop the commercialization of HeLa cells. In 1985, Henrietta's medical records were released to the public without her family's consent. This was another one of the biggest ethical issues in Lax's story. In 1996, the Health Insurance Portability and Accountability Act made it illegal for people such as insurance companies or doctors to make medical records public. The importance of HeLa cells cannot be overstated. Not only did they contribute to developing the polio vaccine, but they were the first cells to ever be cloned or to become immortal. Their characteristics allowed scientists to research cures for many diseases and allowed humans to have a fighting chance in society. Although HeLa cells contributed a great deal to scientific discovery, the way the cells were obtained sparked an enormous ethical debate. Whether or not it is ethical to take tissue from a patient without their consent still remains in question. However, today doctors are required to abide by the informed consent law made for Lax's case.